So I'm actually delighted to welcome you all to the Perkins, and my name is Professor Peter Leedman, and I have the wonderful honour of being the director of this fantastic place. I'd like to begin by acknowledging that the Noongar people of the Whadjuk Nation, the traditional custodians of the land on which we're gathered tonight, and acknowledge their elders past, present and emerging. I would like to acknowledge the Perkins Board Chair. I haven't seen Wayne. There you are, Wayne. Wayne Martin, ACQC. Board members Jan Stewart, PSM. John Barrington, AM. I saw John come in. Roger Port. Saw Roger. Mrs. Bonnie Kirkbride, I caught up with a minute ago. Family and friends of the late Jamie Prinderville, and many warm guests. It's a pleasure to see you all. This is our first Q&A for the year, and of course the COVID-19 pandemic put the kibosh on any of these sorts of events for last year. It also put and delayed the transfer and travel of Professor Jonas Nilsson and his senior scientist wife, Dr. Lisa Nilsson from Sweden, coming here, and obviously we wanted both of them here to have this first Q&A. And I don't want to talk much about COVID. Suffice to say that Jonas and Lisa, we are very pleased that you did make it, because it was a very challenging travel indeed. What I would like to say, though, is what the pandemic has done for medical research is push it and elevate it into a place it's never really been in the cognition of most people around the world. Everyone knows about the development of a vaccine for this virus. Everyone also gets that medical research is the conduit to their health for the first time. And I'd argue that many people take their health and the new medicines that they get for granted a lot of the time, but not anymore. So just before we get into the main agenda for tonight, a couple of very important things. Housekeeping, the toilets are in the foyer halfway down, for those of you who are unfamiliar. If we had a fire, and we haven't yet, but if we had a fire, those doors are going to open and we're going to go out there and we're going to stand and congregate in Verdun Street. Okay, so let's get back to the main game. What are we at the Perkins? We're about 250 passionate, committed scientists from all over the world investigating the causes and developing new treatments and preventive strategies for the major diseases of adults. Cancer is one of our key themes, and I'm sure you all know that, particularly the hard-to-treat cancers. And melanoma, often known as the Australian disease, is a major focus in the Perkins. We are particularly pleased that Lisa and Jonas are here to build their laboratory and their research into melanoma going forward, and I'm sure you're going to hear a lot more about that from Jonas in a moment. But why are we focused on melanoma? Why is it an important part of what we do in the Perkins? And just a very quick snapshot of the facts. It's the third most common cancer diagnosed in Australia. It's the deadliest of all skin cancers. The rates of diagnosis for the disease have gone up dramatically in the last few decades, and it's one of the most common cancers found in young adults. Now, on the flip side, there have been dramatic advances in the treatment of advanced melanoma, immunotherapy and targeted therapies, and I'm sure you're going to hear more about that, which has been transformative for some patients with melanoma, literally transformative, and many of you will know the Nobel Prize was awarded 2 or 3 years ago for the discovery of immunotherapies. It's incredibly important to recognise the support from the Kirkbride family and friends, the Prinderville family and friends, and the investments that that group of people have made into bringing Jonas, an internationally recognised expert in melanoma, to Perth to undertake research here. So it has been a very big community and team enterprise. Jonas's position has been funded through generous donations from the community and from the University of Western Australia to honour the memory of Scott Kirkbride, and we're going to hear a little bit more about that in a moment. Bonnie, you and I have known each other for about 16 years, and it's been an incredibly hard road. But Scott's legacy is nothing short of remarkable. it's already changing the lives of many in Western Australia. So thank you for your part in this huge and evolving story. I think all of you also know that research, and only research, is the ultimate aim that we have for making cancer non-lethal. We won't get there unless we do world-class research, collaborate, work together, and solve the most difficult problems collaboratively. And many of you know, and I'm going to stop talking in a second, I could talk all night about this wonderful place. It is a wonderful place to work, there's no doubt about that. But I'm not going to, because we've got a wonderful panel that Miriam's going to introduce us to in a moment. And that's the most important part of this evening. Miriam Borthwick works in the Perkins now. We're delighted to have her. She's an ex-730 reporter and ABC TV journalist. And she's helping to publicise the work of our scientists. And Miriam, I'm going to hand over to you, and you're going to welcome all these wonderful guests for us. So please welcome Miriam to the stage.
Thank you, Peter, and welcome everyone this evening, and welcome to our Facebook Live audience. It's the first time we have streamed live one of our Q&As, so hopefully someone's out there. I would like to welcome also our panellists this evening. So we have Jonas sitting here in the middle, WA's inaugural chair in melanoma. He was, until recently, the director of a major cancer centre in Gothenburg in Sweden, and has been researching melanoma since 2012 and is regarded internationally as a leader in the field. He's establishing a laboratory here at the Perkins, and tonight we will hear of his work on melanoma, the strides that research have taken, leaps and bounds, particularly in the targeted therapies and the immunotherapy, which Peter alluded to. And for non-scientists, we'll try and translate as we go. On Jonas's right, we have Clare Tate, Dr. Clare Tate, who is the President-elect of the Australasian College of Dermatologists. We're so lucky to have Clare here in Perth. She has worked in the dermatology department at RPH for 20 years, has her own private practice here in Perth, and is the visiting dermatologist to the Kimberley, which I know provides Clare with some quite remarkable adventures in tiny planes, often through cyclone season. Welcome, Clare. And next to me is horticulturalist David Tate, different spelling from Clare, but also originally came from England. David is a 30-year-old melanoma patient and who has very kindly agreed to share the patient experience of having melanoma diagnosis. So thank you very much for being here as well, David. We also have with us, as Peter said, Vonnie Kirkbride, Mrs. Vonnie Kirkbride in our front row. Her son, Nick, who I think will come, so don't worry if someone dashes in, that will be Nick. Many of you will know of Vonnie and Vonnie's family and the tragic loss of her 27-year-old son, Scott, to melanoma. But for those who don't, the Kirkbrides and the wonderful supporters established a legacy in Scott's name, which has had and continues to make significant difference for melanoma patients and treating doctors, and many are grateful to you. So tonight is a very special evening, and I don't usually start with a little story, but I do need to because it is quite a story. So our discussion really begins with the story which is both tragic and remarkable, and it begins with Vonnie's son Scott, a promising golfer who tragically lost his life in 2004. Scott had remarkable parents, and with the most committed of supporters, they set out to create a legacy in Scott's name, the Kirkbride Centre, with the goals of creating a health study, patient database, and to fund a chair in melanoma. And in 2018, so I'm cutting to the chase, moved to the Perkins, the WA Melanoma Advisory Service moved to the Perkins, changed names, so it is now the WA Kirkbride Melanoma Advisory Service, WACMAS. This service provides comprehensive advice to treating doctors and their patients. It's a multidisciplinary panel, what I would call a bit of a one-stop shop, regarding the management of complex advanced melanomas. The aim is to ensure patients receive the best available advice on their treatment and prognosis. And of course, the original aim of establishing a chair in melanoma has been achieved with the arrival of Jonas. So Scott also had an astonishing GP, the late Dr. Jamie Prendeville, and his determination to raise money for melanoma research, well, it was really incomparable. His efforts contributed personally over $200,000 of the more than $3 million that has been raised by loyal Kirkbride supporters. And with a small band of family and friends, Jamie completed 5 ultramarathons. That's 250-kilometre races over 6 days in the Kimberley, the Gobi, Atacama, Sahara, and in Patagonia, these deserts, to raise funds in memory of his patient, Scott. Very sadly, Jamie also succumbed to cancer only late last year. But we couldn't hold an evening like this evening without acknowledging Scott's legacy and Jamie's extraordinary commitment to honour his patient. So we're just going to run a little video now to do that. Thank you, Vonnie, and thank you, Jamie's brother, Gary, family, his partner, Carol, for providing us with those really beautiful images. Vonnie, did you ever think it would come this far?
Not for a moment did we think that we would be in the position we are now. In Scotty's final days, when he was chatting with my late husband, Rob, his brother, Nick, and myself, he said he wanted to, or hoped that his diagnosis of melanoma would help in some way in the future. And we felt we needed to fulfill his wishes and try and do whatever we could, even if it was in a very small way. I realise things take a great deal of time sometimes, and I look back now and I think what's transpired since that early time, Scotty's wish has been fulfilled and we have just come such a long way. That, having said that, it just doesn't happen overnight, and the people that have supported us from day one, believed in what we were trying to do, and I talk about my wonderful family, our dear friends, donors, Peter Liedman, who has become one of my dearest friends, the Perkins, the Prendevilles, the greater community. It just wouldn't have happened without them. They believed in us and have supported us, and I'm so grateful to every one of them. And to everyone here this evening, I think you've not only helped patients, you've helped treating doctors and researchers, and I will always be incredibly grateful, and I thank you from the bottom of my heart. So yes, I guess I had no idea what we were letting ourselves in for, but I knew we had to try and do something, and you know, Scotty was an extraordinary young man and still miss him terribly, but I think we've hopefully somehow helped others, and so thank you to each and every one of you for being part of that and getting us to where we are today.
It has, and it is a remarkable legacy. So well done, and thank you for being here this
And may I just say one last thing? Jonas, welcome to the Perkins. We're so excited to have you here with your family, and I wish you every success and loads of happiness here in Perth. It's a pretty good place to live.
Yes,
So we're thrilled to finally get you here, so thanks so much.
Thank you.
We've just been hearing about Jonas and his family's delight at discovering open-air cinemas.
Great. Yeah, probably something you wouldn't get in your part of the world.
So, Jonas, let us start with you. It is quite a backstory to your arrival. So much has changed in the world of melanoma since Scott's death in 2004. How far has it come?
I think it's a remarkable time to be a melanoma researcher at the moment, because we have really been able to see what basically basic research can do for a disease that very few ever thought was possible to treat. So many of my colleagues a long time back said that people should work on melanoma if you're really clever, because that's the most difficult one to treat. And now I think what's remarkable with melanoma is it's actually the poster boy for two types of treatment, both recognizing how the genetic material in a cancer cell is changed, as well as understanding how immunology works, has really given so much to this treatment arsenal, which we probably never thought around 2004 to be true like it is today. So it is a remarkable journey.
Yes, and one of the two treatments we'll talk about a little bit later, immunotherapy and targeted therapy. Yeah. David, immunotherapy, one of the new discoveries, has been part of your treatment. Now, David's melanoma story, if I'm correct in remembering, 2015 you noticed a mole changing shape and colour on your leg, went to see the GP. That resulted in a biopsy which confirmed melanoma. Lymph nodes were removed, regular visits to dermatologists, scans, went on until 2019. You were probably thinking, this is fine.
Yeah,
it's not happening. Fine. And then what did happen?
So as you say, I had my yearly PET scan every year. Everything was fine. Saw the dermatologist every 3 to 6 months. And then January 2020, I had my routine PET scan, and I said to my dad, I can't believe I'm having another day off for this PET scan. But it showed metastatic melanoma. So there was widespread disease through my gut and,
most of your torso, your lungs and,
So that was quite a shock. So I saw the oncologist and he got an MRI, got me to do an MRI, and it showed of the brain. And I had a small lesion on my brain. So the first step for me was to have a craniotomy to get that excised, which went very well. And then I was going to do a trial, and it was going to be pembrolizumab and a trial vaccine. I had to wait 28 days after major surgery to start the trial.
Those 28 days must have been your hardest.
Probably the hardest throughout the whole journey, I'd say. Because during those 28 days, I had other symptoms appear. So I had lumps appearing.
On your body?
Yes. Yeah, so on my back. So that was quite difficult. And I did a CT scan in preparation for the trial, and that showed progressive growth in the tumours. So, and I had this other symptom which was quite difficult.
I mean, this is really illustrating how rapidly this disease was moving.
Well, I had the brain surgery within about, it was within 2 weeks of finding out.
And so this was also at the time of COVID, and you had COVID-like symptoms, so then that needed to be tested for as well.
Yes.
And you also are a type 1 diabetic. So you really, you know, lots of people said 2020 was a shocker of a year. It was an absolutely ghastly year for you.
So the COVID symptoms arose because I was pulled out of the trial and I started combo therapy, Opdivo and Yervoy. So I did 3 cycles. After the 3rd cycle, as you say, I started to feel unwell, and so I called my the oncologist and said what my symptoms were, and of course they were all
the
matched.
same symptoms as COVID. Yes.
I had flu-like symptoms, temperature, so they got me to go to hospital, and I had to wait to get the test results of COVID. So I was more worrying about the test results of COVID because I was thinking, what would I tell work? Everyone would have to go home for 2 weeks. So I was quite anxious about that. But obviously I was negative. But it was just the liver reacted with the immunotherapy.
Yes.
So
Now, Claire, I just want to say that, I mean, sadly, melanoma is a disease that can and does affect young people as well. Can you just briefly describe, we've just been hearing all of these symptoms that David has gone through, and they have been really rough. At a cellular level, so if we have the mole at the surface, in the skin cells underneath that surface, what is happening which can lead to it being a metastatic cancer?
So when a melanoma starts off, the cell it originates from is called the melanocyte, which is the pigment cell in the skin. And they are scattered throughout the very top layer of the skin. And as a melanoma develops, those melanocytes become abnormal and they multiply. And initially, they just multiply in the very top layer of the skin. And that's what we call a melanoma in situ, or a level 1 melanoma. And because there are no blood vessels or lymphatic vessels in the top layer of the skin, the melanoma cannot metastasize. So that's why our great responsibility as doctors, and particularly as dermatologists, is to detect a melanoma at that very first stage when it's just located in the epidermis, the top level of the skin. As the melanoma develops, it drops abnormal cells down into the second layer of the skin through a very important structure we call the basement membrane, which is usually a very good barrier. But these clever cancer cells get through that basement membrane and drop down into the second layer of skin where the blood vessels and the lymphatics are. And as soon as they're there, there's the potential for metastatic spread. And the lower the melanoma cells go in that second layer of skin, and sometimes into the third layer of skin, which is the fat layer, the bigger the blood vessels, the bigger the lymphatics, and the greater the chance of metastasis.
Great, thank you for that. And Jonas, we've talked briefly about that really when you began researching in 2012, life expectancy for someone with metastatic cancer was very low. Yeah. But then came along the targeted therapy and the immunotherapy. Can you briefly, simply describe those?
Yeah, so I mentioned before that there are changes inside of a cancer cell in the genetic material. So you know every cell has DNA, which is the genetic material. And if you get changes in certain points or places in the genetic material, this can result in that the cell starts to activate certain signaling events inside of the cell so that the cell starts to divide uncontrollably. And around 2002 or so, they discovered that these alterations in the genetic material actually was encoded into a protein called BRAF. And this protein then becomes overactive and starts to telling the cell to divide. So then the obvious idea would be then if we can find a way to generate a small molecule that can bind to this activated protein and stop it from telling the cell to divide, then we would have a therapy. So they made this, and it was remarkable responses in cultured cells and in animal models, and then they tried it in patients, and there were case studies where patients that had metastasis all over the body got this pill with this BRAF inhibitor, and the tumor just melted away. But there was a problem. Unfortunately, within 6 months to a year, the cancer came back. And this was then because the cancer cell had figured out, okay, if I cannot use this BRAF, how should I do then? Well, they started to activate other types of signaling nodes inside of the cell so that the cell started to divide again. So ultimately, even though there were huge therapeutic responses and actually delayed and prolonged survival with this targeted therapy, there was no cure. At the same time, completely independent of this understanding of the genetics of tumors, there were also people interested in identifying how the immune system can recognize cancer cells. Because there had been case studies where people had said that they had a melanoma and all of a sudden it was gone. So we knew that somehow the melanocyte or the melanoma can be detected by the immune system somehow. But we had never really figured out how we can get many patients to benefit from the immune system. So then they identified certain molecules on the surface of both T cells, which is a critical cell inside of the immune system, and on the tumor cell, which made an interaction. So as soon as an immune cell comes in and wants to kill the melanoma, then since the immune system is like the police, the tumor cell quickly puts up a donut on the surface.
A donut.
A donut. Yeah, and it really looks like a donut, and we all know that cops love donuts. So they start to eat this donut and they sit idle next to the tumor cell. So then they figured out how to get rid of the donut by generating antibodies that can go in and hit the donut, and all of a sudden there are immune cells that are sitting next and eating the donuts, and all of a sudden there's no donuts, and they get really angry, and they can then kill the tumor cells. It sounds funny, but this is actually the way it is. And you can have remarkable responses to blocking this donut, which is then called PD-L1, and the binder of this donut is called PD-1 on T cells.
And so can this treatment, or the combination of those two treatments, can actually some patients be cured?
So, we're generally careful about using the word cure. If you think 5 years after complete responses regarding cures, then probably there are quite a few that has been cured by immunotherapy predominantly, but targeted therapy, extremely few have been cured by, if any. Mm-hmm. So we actually had in Gothenburg a patient that after 8 years on a complete response with targeted therapy got the cancer back. And this is not what you see with the immunotherapy. There, the durable responses can very much look like cures. So we are very hopeful that immunotherapy can cure a fraction of patients at least, currently around 20 to 35%.
That's not a fraction, that's quite a good size.
Then how many respond is a lot more.
Yeah.
So
David, how's the treatment been for you? How have you, you talked earlier of having the lumps appearing, did they, as Jonas used the phrase, melt away? Did you find that you responded well to the immunotherapy?
So within a week of having my first treatment, I noticed lumps disappearing and replaced with bruises. So it was as though they were exploding. That's how I pictured it. I didn't have, when I found out I had the metastatic melanoma, I didn't feel unwell. The oncologist rang me up when he found out and said, look, how are you feeling? I said, just a bit tired, because I work outside in quite a physical job, so I just put it down to a long, hot summer. But I was doing quite well up until the third treatment, and then that's when I started to feel unwell, like physically. So it was actually the treatment that was making me unwell.
But the tumours have responded well to the treatment.
Yes.
Yeah. Clare, David's job was as a horticulturalist at Kings Park, and as we can see, red hair, pale skin.
not a winning combination.
Would his skin type, or people with similar skin type, is that a determining factor?
It's one of the risk factors, certainly. So there are sort of 5 or 6 main risk factors for the development of melanoma. And David has what we call type 1 skin, which is the very fairest type of skin it's possible to have. And we know that all skin cancers that we see commonly in Australia, and certainly melanoma, are much more common in people of fairer skin type and are really uncommon in very dark skin types. So that's a definite risk factor. There are other risk factors as well. So the other one that you have, David, is working outside. Occupational sun exposure is a definite risk factor.
What about genetic predisposition?
So genetic predisposition is very important, and if you have a first-degree relative, so that's a parent or a sibling or a child who's had melanoma, you have an increased relative risk of about 1.7, so that's also significant. There's a condition called dysplastic nevus syndrome, where an individual has more than 5 unusual-looking moles that are not melanomas. That's an independent risk factor. And total mole count, or total nevus count, is also an independent risk factor. So some individuals have more than 100 moles, and that is also a significant risk. And then if you've had what we call a non-melanoma skin cancer, so those are the commonest skin cancers. The 2 commonest are basal cell cancer and squamous cell cancer. If you have one of those before the age of 40, then your risk of melanoma increases.
Right.
So we're very keen to encourage anyone in those risk groups to actively have regular skin checks.
But what should we all look out for?
So
we've all got funny coloured bits.
Yeah. So we're pretty simple people, dermatologists, and it's been broken down into an alphabetic mnemonic that helps. So A stands for asymmetry. So if a mole is growing and looks very asymmetrical, or has previously been symmetrical and becomes asymmetrical, that's a cause for a consultation with your doctor. B stands for border, so we like moles to have very nice, regular, round borders, and if it starts to look a bit what we call geographic, a bit like a coastal inlet with bays and variations in the outline, then that is also something that should be checked. C stands for colour, so most moles are just one colour. They might be light brown or dark brown or black, sometimes blue. But if you start developing different colours in a mole, and those colours might be white or red or any shade of brown or black, that should be checked as well. And D stands for diameter. In the old days, we used to say any mole greater than 6 millimetres, but now we want to pick up melanomas very early, so D stands for increasing diameter. If you have a mole that is growing, it should be checked. Right. And then E is evolving, so a mole that's evolving or changing. Really important. Now, all those things can happen, and it might not be a melanoma, but all those changes are a signal that you should see your GP and have a check, and if your GP is worried, they can refer you to an appropriate specialist if needed.
David, when you first noticed the mole on your leg changing, what did you notice?
I noticed increase in size. So it was always a flat, round mole. So it increased quite a lot, and the edges became sort of scabby or red, I would say.
And over what period of time?
About 2 weeks.
Oh gosh, really quick.
2 or 3 weeks. I do live with a certain amount of regret in terms of possibly not getting it checked quicker. That does sometimes. I do sometimes think about that, but it was about 2 or 3 weeks.
Okay. Now, Jonas, when you talked earlier about the targeted therapy and the immunotherapy, but of course cancers are always very clever and they're always devising new ways to survive, which brings us to the need for a new breakthrough, and you being here wanting to set up a biobank. Can you tell us a little bit about what a biobank is and why you want to establish one?
Yeah, so a biobank is basically tissue collection of tumors and blood and other types of liquids one can collect, and then do analysis of. And we are particularly interested now that we have these two types of therapies, targeted therapy and immunotherapy, to do a biobank of materials that comes from patients that do not respond to these diseases. Because now it is actually standard therapy, so every patient will eventually get these types of advanced treatments. So it's not like it used to be, where you had to be connected to a clinical trial in order to get material to investigate what are the resistance mechanisms. So that's what we're interested in now, figuring out why do some patients respond and why do others. And out of our lab kind of specialization is not only to look at the disease, but also try to do something about it. So if there is a patient that has a resistance development, we would like to try new ways to attack that particular person's disease.
Yes.
And not just only wait until we have collected a large biobank, but also start to analyze different new ways of targeting the disease.
Because there would be biobanks around the world, so why do we need another one?
Yeah, it would, actually two different reasons. First, as I mentioned now about the standard therapy. Second, also what we've noticed when we were back in Sweden was that there was a lot to benefit from having local biobanks, because it's local patients. And if you happen to be serendipitous in your findings analyzing the biobank, you could in principle envision having real-time data that can be brought back from the lab to the clinicians and actually guide treatment of the very same individual from which the biobank's tissue was generated. So that's happened a few times in Sweden, where we were trying to generate advanced animal models based on how the tumors looked in a particular patient. And at sometimes it just happened to be that when we figured out why the tumor was resistant and ways to target it, it actually could benefit these patients. So that's why I think it's not only for the future, but also what can you do when you have the patient in front of you using the hugely advanced technology that is right now here at Perkins, with a new genomics facility and also equipment that Peter and others have brought in to this institute.
That sounds very promising. Claire, I want to come back to the patient perspective. Peter, in his introduction, said that the rates of melanoma are not declining, they've actually increased. We've also, though, had, 40 years of, I think I was young when slip, slap, slop, we were all told about. So has the sunscreen method and campaign not worked?
Well, it is very interesting. If you look at age-standardised incidence of melanoma, particularly over the last few years, there is now a reduction in incidence in melanoma in the younger age groups. We're seeing that for the first time.
So it's an accumulated effect over life.
It is. So the risk of melanoma increases with age. So the older we are, the more likely we are to have melanoma. So that may be partly why we're becoming an older population. But actually looking at the younger ones who've had the benefit of this quite extraordinary public health campaign that we've had in this country now for about 30 years, I think, we're just beginning to see benefits of that.
Right. So long-term public health campaign.
Very important, yeah.
Okay. Jonas, you actually, I believe, tried with one poor mouse and shaved half of it and put sunscreen on it. How did it fare?
Well, it's not our work. It was done by a group in UK, because actually most of the evidence about sunscreen have come from epidemiological studies on basically the population. But no real strong experimental evidence of the benefit of sunscreen had been done. So basically what they did, they had a mouse that had been genetically changed so that it will develop melanoma at one time point during its life. And then they gave them basically UV radiation after having shaved the fur away from the mouse. And on one mouse they gave sunscreen, and on the other one they didn't. And they could actually see that this reduced the melanoma development in these mice. So that's kind of the first experimental evidence. And that was really, really late, just less than 10 years ago. So although we've talked a lot about that it should be protective, sometimes the criticism has been there are only epidemiological evidence, but now we have pretty strong as well experimental evidence that the sunscreen does change. But they had to use 50 in SPF. Anything less is not really working.
And the other interesting side to this is that there are different types of UV radiation, and the UVA and UVB are what we are exposed to. And traditionally, people always assumed that it was the UVB that was the real culprit in melanoma development, but we're now getting increasing evidence that UVA is also really important, and that may be through UVA's effect on suppressing the skin's immune system, which it does very effectively. So sunscreens are now being developed with what we call broad spectrum activity, so activity against UVA and UVB, and that's really important. So buy, as Jonas says, a 50+ broad spectrum sunscreen, and you'll be very much better off.
We've also seen the recent popping up of mole scan clinics, sometimes caravans, even on the beach. Is that as effective as going to see your GP or a dermatologist? How would you rate them?
It's a really difficult question, because you're asking me to rate individuals who are working in different settings, and I obviously don't have individual knowledge of those individuals. But as a general rule, my advice would be always see your trusted GP, because they know you, you know them, and all good GPs know their own level of experience and expertise. So if they feel they're confronted with something that they are not capable of dealing with, they will refer you to a specialist they know and trust. I think that's a much, much better pathway than going to a non-accredited clinic. My concern about the skin cancer clinics and the mole clinics is that there is no standard of care. They're not accredited by anyone. There's no particular training that the doctors have to do. So you may chance upon someone who's very, very good, who's taken it upon themselves to do lots of training, but you might not, and you really have no way of knowing. Yeah, it's a difficult question.
I suppose it raises awareness, if nothing else.
Yes, and I suppose this also leads on to, should we be doing screening of populations? If these clinics are cropping up, should we be encouraging everyone to go? And it is interesting because the Cancer Council at the moment in this country and in the US do not advocate population-based screening, because they feel there's not enough evidence that it reduces the death rate from skin cancers, but we strongly advocate what we call opportunistic screening, which is if you have the opportunity to have a skin check, have one, and particularly those individuals in those high-risk groups that we talked about earlier with those particular risk factors. And also, everyone can do a self-skin examination. We can all learn to look at our skin, to know what our skin looks like, and then we'll hopefully discover if there's a change. And a significant number of skin cancers are picked up, as David, you picked up yours, which is really fantastic, by being aware of what our skin looks like and noticing a change.
Or even barbers and hairdressers, I believe.
Absolutely, yes. Actually, a lot of people come in saying my hairdresser was worried about this, and that's fantastic. So the more we increase awareness, the better we all will be.
Jonas, you talked a little bit earlier about that very individualised approach of sort of research-patient link while the patient is still alive and finding a successful treatment. Now, I know you've come to Perth not only with your wife, Lisa, and I've been dying to say this, Lisa is a mouse avatar expert, and a couple of other members of your team. So can you tell me a little bit about that work, which is really the personalised, that connection between the patient and the research?
Yeah, basically, mice have been used in cancer research for basically over 100 years and have been instrumental in really understanding how cancer develops, and also as models, if you want to try a new treatment in patient, you always have to try this first in animal models to know that it's safe, etc. So what we developed a few years back was the idea that perhaps we could have one mouse per patient, and where we would basically make a human-like mouse that we call a mouse avatar, or a patient-derived xenograft, which is basically that inside of the mouse the human tumor can grow, and we can treat this mouse then with different types of treatment, and then see if there is treatment responses. Then we would know that this patient could potentially get this treatment.
That sounds like something you would want to do with all cancers.
Yes, but the problem is that there is not so good success rate in generating these types of animal models for other cancers, except melanoma, because melanoma is just have a very aggressive growth pattern, and they can grow under the skin of the mouse and form tumors. And the mice basically don't get so affected by it. It just becomes a lump, and they can grow to quite decent sizes. So we can start to measure them and to treat them with different, without overtly having big effects on the well-being of the mouse. So they become kind of culture vessels for human tumors, if you like.
And you've actually had some results this way?
Yeah, so the first results we got was, of course, in targeted therapy, but then came immunotherapy. And in order to grow these tumors inside of the mouse cannot have an immune system. Otherwise, it would have reacted immediately to the human tumor, and we wouldn't have any model. So when immunotherapy came strong, we set out on a mission to actually humanize the mice, so that the mouse didn't only have the tumor from the patient, but also immune components. So that's what we were the first lab in the world to be able to develop a model where we could look at the interaction between a human T cell and a human cancer inside of a live animal. Everything all works before had always been in mouse tumors with mouse immune cells. Here we were able to study inside of an animal. And that actually gave us two ways. First, this new model, but also secondly, a new way of attacking melanoma cell, which is called CAR-T therapy, which is a chimeric antigen receptor therapy that has previously been used in leukemia, very successful, but there's so far no CAR-T therapy developed for melanoma. But in our animal model, we were able to actually develop it further and use this new concept of immunotherapy, which is basically growing immune cells outside of the patient's body, genetically alter the T cells so that they can now recognize a melanoma that they normally do not recognize.
So it's almost like a numbers game. If you can grow those immune cells outside of the body, you can actually bombard the cancer. Is that the thinking?
Yes.
Okay. I do want to leave some time for questions from the floor because we are running out of time. So I'll ask one more question while you all think of what you would like to ask. David, are you still working in horticulture?
No, so late last year I made the decision to resign from my position. So I'd spent 7 years working there. And I just thought the treatment was going well. I'd had a surgery a few months before that, which went well. And so I just made the decision just to chill out over summer and put my health first. And I made a good decision. I actually have a job interview tomorrow, doing a similar type job at another botanic park. So fingers crossed. Because I do feel now ready to go back to work. Everything's going well. I'm still responding to the treatment. There's a lot less burden on the body, so I'm happy to go back to work.
Well done on taking that period of time. Yes. Now, Lydia down here has a microphone, and there's one up the back too. So if someone would just like to raise their hands, because it's much easier if we use the microphones. Are there any particular questions people would like of any of the panel? Over there, Kirsten, and thank you.
A question for David. How big was your mole or whatever it was that you started off with that became the melanoma?
It was about, it'd grown maybe maybe quarter of a centimetre raised. So the melanoma, I do actually have the measurements here. I've brought it with me. So it was, 2.47 millimetres, and the Clark level was 3/4. I don't quite know what that means. I think
diameter, circumference.
I think that measurement there is depth,
Yeah,
I believe.
thickness.
Yeah, the thickness. So I think it raised about that much. How much? That's like
2 or 3 millimetres.
2 or 3 millimetres raised, and it also immediately before I had it removed, it developed a sort of round red outline around the mole. So it didn't change colour. It just started to maybe look a bit scabby around the edges, and that was why.
Thank you. Now, there was another question near you, Kirsten.
I just wanted to ask about internal melanomas, because I understand that they can develop inside the body and that sometimes people can have metastases from melanomas, and they don't ever discover where the original source was. So could you answer that, maybe Jonas?
Yeah, so actually skin melanoma is by far the most common one, obviously, but there are other forms of melanoma, including mucosal melanoma, which is melanoma that is in the mucosal areas of the body. You have also a very aggressive form of melanoma called uveal melanoma, which is an eye melanoma that actually our research group is very, very much interested in. It's about 2% of all melanomas, so not often, it's quite rare, but it is very lethal with about 50% survival rate, and it's very interesting biology, completely different than skin melanoma. And then what I think maybe you are referring to is a cancer of unknown primary, basically, that where there has once upon a time been melanoma somewhere in the skin, but you just never spotted it. But because the melanoma cells, they just went and they metastasized before there was a really, really visible melanoma. So that can happen sometimes. I don't know how many percentage, but it's not rare. It does happen.
We do see it in clinical practice, yes.
Yeah.
So skin cancers can undergo a natural phenomenon known as regression, where the body's immune system, which we've already talked about, attacks the tumour without any help from treatment. And so sometimes a patient can develop a melanoma that they don't realise is there, and then the body's immune system comes in, it regresses, it disappears, but before it regressed, some of those cells had slipped down into the deeper layers of the skin, and some of the cells had got into the blood vessels and it had metastasized. So that's probably the commonest cause of what you quite correctly call an unknown primary. Yeah.
One last question. I'm sorry, we're going to have to keep it tight. I think you leapt in quickly. Well done, yes.
I'd just like David to say a little bit more about the lumps that he developed. Was that associated with the melanomas or with the immunotherapy?
So the lumps were subcutaneous melanomas. So they were just under the skin lumps that started to appear before I'd started treatment. So once I'd started treatment, they were the first sign of response, because I had a few on my bottom that started appearing, and so I felt them when I was sitting. But once I'd started treatment, they were disappearing.
It's quite remarkable how quick that has occurred. We are going to have to wrap up our evening, but thank you for all coming, and I hope it has been a really rewarding and interesting evening.